Good morning, dear family. After 1 week of break, because of the convocation, we come back as a family to conduct and have fellowship under this Quest banner. And today we have an international speaker in our midst, an erudite scholar. And to welcome him, to introduce him, we'll have our moderator for the day, John Valliattu sir, doing it. And before we start the proceedings, may I request our own Marina Thomas ma'am to start with a word of prayer.
Almighty God, loving Father, thank you, Jesus, for this beautiful day, for this opportunity thou hast given us to assemble here, Holy Father. Lord Jesus, we pray for the speaker today, Dr. Libin Abraham, Holy Father. Thank you, Master, for bringing him here to share his wealth of knowledge with us so that we can gain knowledge and help our patients here, Holy Father. We pray that whatever the speaker tells and from his experience is useful for us, and we are all benefited and blessed. Thank you, Jesus. We ask all this in the precious name of the Lord Jesus Christ. Amen.
Thank you, ma'am.
Good morning and welcome. I think because of the rain, our audience is small, but we are going out live on YouTube. So I hope we will have a YouTube audience, and also it will be there for us to watch later on. It's my great pleasure to introduce a very unique speaker today. Libin Abraham is married to my niece, Veena, my brother's daughter, and both of them together did the MSc in biotechnology in India, and both of them went to Germany quite a number of years ago, and both did their PhDs from the University of Heidelberg in Germany, and then both of them went to Vancouver, where they did postdoctoral fellowships in cancer research. And Libin now is working as a senior scientist in T-cell immunology at Therapeutics, Canada, Vancouver, Canada. And Veena also is, her field is what is she doing? Cancer, yeah, she is working on cancer genetics. They are full-time researchers, and they are right in the middle of it. And what you're going to hear today is Libin's and his colleagues' own work, and he's extensively published, which I don't have a list of that, so I'm not going to reiterate to you. This is, as I said, straight from the horse's mouth. He's right in the middle of it. So this will be an authentic speech, I can guarantee you. With great pleasure, I introduce to you Dr. Libin Abraham. Libin.
Good morning, everybody. It's truly an honor, and I'm very excited to present my data today because it's the first time I'm presenting my data to a different audience. Because every time I present to my colleagues or when I go to conferences, I present to researchers. But this is the first time I am presenting my data to doctors like you. And I'm truly humbled, and thanks for coming today. As Dr. John Matthew has already given the introduction, I'm going to touch a few aspects of cancer today. And I will promise that I will not confuse you with any of these long acronyms or unfamiliar words. If you feel that this is not clear, please feel free to interrupt me right away so that we can have a good discussion towards the end. So for today's agenda, I just wanted to obviously start with cancers and some of the major risk factors we have for cancer. All of us know all this, but I want to touch base on that because that's a very important requirement to set the stage. After that, I will be talking about two major cancer immunotherapy tools. The one is called BITE, again, bispecific T-cell engager. Some of you might be familiar with this. Another one is the cell-based therapies. So the reason I'm talking about BITEs and cell-based therapies is because, as again some of you might be familiar, this is one of the top 2 leading candidates for cancer immunotherapy now. The next 2 sections will be mainly about my work, which I did during my postdoctoral work. I collaborated with a company and who was actually developing biparatopic antibodies, and I'll come to that all. And this is now in the phase 1 clinical trial. I'm very excited that I was able to work together with them, and let's hope that this brings some comfort to patients. And the 4th topic is about my current research in my company, which is induced pluripotent stem cell derived CAR T cells. And again, this is one of the most fascinating research I have done so far in my entire career, because truly this is the next cutting edge technology and the science we have. And I want to end the discussion today with some of the challenges we have today in the CAR T cell therapies. So, I'm not going to define what cancer is, and we all know what cancer is. This is one of the major leading cause of death. According to the CDC, or the Global Burden of Disease 2017 report, this is the 2nd most leading cause of death. But what I want to really emphasize is that, I think there is a common misconception that, most people think that all cancers are same. And this is somewhere, it's very tough for, again, doctors like you and also, science communicators like me to really tell people who actually have been in touch with cancer that they're all very different. And depending upon the type of cancer, the staging of the cancer, grading and different criteria, and also the person's own immune system, you might have a different outcome. So I just showed here the 2 pie charts where you can clearly see that, they took it from the World Health Organization, and we have the major causes of leading cases from the breast and lung cancer, but we have much higher mortality mainly from the lung, and we have much more decrease from the breast cancers. The colorectal cancers is still very, very challenging. So if you now look on this data set, and I got this from the American Cancer Society page. So this is a US data set. I wish I could show something similar to you from India, but I really like this data because they have captured a lot of information. So the first thing is the color codes, you're seeing what is the age at which these diagnoses are made. You can see that most of the cancers are actually color coded in this color, which means they're all on people who are above 65 years of age. And you can see also different types of cancers and what is their 5-year survival rate. And I highlighted a few things here because you can see that the pancreatic cancer, which has a very poor outcome, 7% survival in 5-year period. On the contrary, some of the other ones, for example, prostate or larynx has higher. The reason I emphasize these 2 is because we have a very famous politician who passed away because of pancreatic cancer. And we also have a former chief minister who is now struggling with cancer. And people keep comparing, why is one person's cancer so aggressive? Why is other person's not? So I really wanted to take a moment to appreciate that we have different kinds of cancers. They come at different, the onset is very different. And their 5-year survival is also very different. And this is very important as doctors and physicians, clinicians like you, to understand how you will be treating these patients. So what is the cause of the cancer? And I'll be only touch on this very briefly because this was an extremely annoying report which came. And I think most of you would have heard this in 2015. It came all over the news, and BBC actually wrote an article like this. Most cancers are just bad luck. And look at the picture they gave: cigarettes, alcohol, and maybe a remote control, just to show that it's less physical activity. And they say that this is published in Science, one of the highest, or one of the most reputed journals. And they show that 2/3 of the cancers were just by chance mutations or stochastic mutations, and you don't have any control on all cancers. And that was very bad, and even it came in Forbes. And this study came from Johns Hopkins, and I hope most of you know that. And they say that bad luck or random mutations play a major role in this. So I know this was very, very bad communication. Most people who are heavily drinking, smoking, having very bad life habits, they were excited to see this news because they were like, okay, now I can start smoking, now I can start drinking, because it's just a bad luck. So just tossing a coin. So what exactly is that paper? In that paper, what they did is that they actually have a plot, and this was a major plot which they took to come to a conclusion. And you'll be surprised how bad the conclusion is. So shown here is different type of cancers, and on the x-axis is the stem cell division, so the number of times the cells divide. And on the y-axis is actually the lifetime risk. So the long story short is the more times the cells divide, they acquire totally random stochastic mutations. So they made a conclusion that, okay, everything is just stochastic, everything is random, and it's just a bad luck. It's called the bad luck cancer hypothesis. So this was so bad, World Health Organization has to come forward, the International Agency for Research on Cancer, and they had to really say, we strongly disagree to this, and please follow all the recommendations, please don't smoke, or please don't consume alcohol, and all other risk factors which causes cancer. And they really actually condemn this very badly. And the good thing is that science should be reproducible. That's a good thing about science, right? When you do science, you need to have reproducible data. And another group of researchers took the same data sets, and they found that the previous research was right. Actually, that is actually the basal rate. So highly dividing cells have high cancer occurrence. That's the basal rate. But if you look all the blue dots, especially the lung smoker here versus the lung non-smoker, you can see there's a big difference. So they say that the conclusions might be same. You have high incidence of cancer depending upon cell divisions, which is a basal incidence of cancer. But on top of that, you can increase your cancer risk depending upon your habits. So they clearly told that we have a lot of extrinsic factors like your diet, your lifestyle, which is also very important for the cancer risk. So I'm not going to go to any of these details, but all of you know that we have a list of things which is actually again provided by the Global Burden of Cancer published in 2022 actually in Lancet. So you can clearly see that the contribution of all of these factors and the different type of cancers. I really don't know how controlled are the normalizations, but at least we can appreciate that the top three, like the tobacco, alcohol, and dietary, is still a major factor, and we should really, communicate this as much as possible and tell people that, you're putting yourself into more risk by these habits. With this said, now I can start transitioning to cancer immunotherapy. So I don't know how many of you are actually working with cancer immunotherapy, or how many of you just been familiar with this. But the whole concept of cancer immunotherapy is that we are utilizing the components of our own immune system so that we can fight the cancers better. So that is that beauty about cancer immunotherapy. So in this schematic, I have shown here three basic subdivisions. One is a cell-based therapies. So here we are using cells, which are engineered cells to kill cancers, which is actually called the living drugs. So technically, it's not like taking an acetaminophen or any other pill. Rather, these are engineered cells in your body trying to find the cancer and selectively killing them. And there are different types of this, which I'll come into that in a second. Second, these are antibodies, and I think biologics, which most of you are familiar with, immune checkpoints. And we have a lot of these ones, the PD-L1 and CTLA4 and so forth. And the third type is actually a combination of many things like the cancer vaccines, bispecific antibodies, cytokine therapies and so forth. So I'll be going a bit in detail about bispecific antibodies because this is actually getting super exciting in the field of immuno-oncology. I think I should share some of the excitement things happening here. So what exactly is bispecific T-cell engager, or BiTE? So shown in the top scheme is actually, I'm taking you back to the basics of immunology just to brush a few things. So you can see, we know that we have a T cell here, and here is actually an antigen presenting cell. So we know that always the T cells interact with an antigen presenting cell, which will be carrying a peptide. It could be a self peptide, it could be a foreign peptide, viral peptide, cancer peptide, and so forth. And then this is how this synapse is getting activated. But what happens in BiTE is that we are creating this kind of synapses or contacts by using a bispecific molecule, which is actually coming from antibodies. So you can see here that it kind of very much similar to that. And if I zoom into what is happening here, you basically have two antibodies here. One is an antibody for T cell receptor, which is a component of T cell receptor called CD3s. And you can take the top part of that antibody and you can engineer it. And you have another part which is specific for a tumor antigen. So what you do is that you have antibodies which are raised for both of these, and you can take the top part of it and then fuse it together to form a BiTE. And this bite is so good because you can see that like they really bring it. So the purpose is that like when you have a cancer and you have a cancer antigen on the surface of the cancer cell, you can recruit any of your T cells into that and make it kill the cancer. So you are actually not having that specificity of T cells come into play here, because we all know that we have a repertoire of T cells. We have a T cells for viruses, for coronaviruses or other viruses or infectious diseases. We have a large repertoire of T cells and TCRs. But here, we are actually bypassing all this by just getting any functional T cell in our body to come towards the cancer and kill it. So that's really beautiful. That's really nice, because this is the first time we are able to really utilize our immune responses and make them come towards a cancer and cause a targeted killing. And why bites is because, as I told you, like you are engaging patients' own T cells with the tumor-expressed antigen and thereby eliminating the cancer. It's off-the-shelf product. Like once you make a bite, for example, you have a bite for example, a CD19, for instance, like a B-cell leukemia, or some other antigen cases. You can actually have it in the, just like how you go to the drugstore and buy a pill, you can have the bites. And it only takes a clinician to administer this through IV. So it's very good. And there is no need to do lymphodepletion for these people. It has a very good safety profile and with minimal side effects. And one thing about the bite is that it has a short half-life. It's just a protein molecule, so obviously proteins get degraded. And in a way, it's kind of nice. You might need multiple infusions, but at the end of the day, the clinicians have a very good control of using the bite, dose the bite, and accordingly get the clearance of the tumor. So bites are actually getting very popular now. And I want to take a moment to explain how this field is exploding with a company called AbCellera. So some of you might have heard about AbCellera because when COVID came, we were looking for COVID antibodies, monoclonal antibodies for COVID. And this was one of the first companies in the world which came with an antibody specific for coronavirus. They were able to do that because they have a technology where they can identify a needle from a haystack. They have this single cell screening platform by which very rapidly they can actually go through your entire immune repertoire and get the B cells and the correct antibodies, which will be the most effective ones. So they actually got blood from a patient who actually got naturally infected with coronavirus and recovered, so that he has B cells which make antibodies. Took the blood, immediately screened it. In less than 90 days, they could actually build the whole antibodies available for the patient. So this is actually a really powerful platform, and no other, let me emphasize, no other technology in the world today can do this faster. And even now, if you look at the best monoclonals available for COVID, it's actually from AbCellera, because they have been continuously iterating it for Delta variant, Omicron variant, and so forth. So what do they do is that once they have the antibodies from your own immune system or our own immune system, it's not only just identifying the needle from the haystack, they have the tools to sharpen this needle. And they sharpen this needle because they want to really make sure that this is the most effective antibody for cancers. So I would really encourage all of you to look into AbCellera, because in future, just like we talk about Roche, Novartis, Johnson & Johnson, AbCellera is going to be one of the biggest antibody players. And we might be one day using AbCellera products in this hospital. What is unique about this is that they already have a product. They have 7 in clinical trials now. But if you look at the structure of these antibodies, again, it is a bite, so it has 2 different specificities. One part binding for the T cell part, which is CD3, another part binding for the tumor antigen. And if you look at this curve, if you can see that all this, this is a tumor killing. And you can see that all these open triangles, you can see that the tumor is nicely killed. And, sorry, the other way around. The open triangles, the tumor is nicely killed. You can see that there is more killing of the tumor, and the T cells are nicely activated. So this clearly shows that how powerful is a bite for really killing cancer. Again, I wanted you to go home with this wonderful experience that you are using any of the T cells in your body to kill the cancers. So the second thing I want to talk about is another work which I did during my postdoc, and I'll be spending a few minutes about this. This is actually a collaboration I did with a company called Zymeworks, again based in Vancouver. And here they were doing something very similar to bites, but not very exactly the same, because we are not engaging a T cell, but the antibody design is like a bispecific one. So it's called a biparatopic antibody against a molecule called HER2. And most of you might be familiar that HER2 is very common in many cancers, breast cancers, gastric cancers. So if you have a HER2 high cancer, then, and if they are responsive for antibodies, then you need to get the best out of it. So we published this paper early this year, and one of the unique things is that currently the best available antibodies, molecular antibodies for the HER2 is the trastuzumab, which is called the TRAS, and the pertuzumab, which is called the PER. And both of them bind to different domains of this HER2. But the way this antibody is engineered, you can clearly see that antibodies don't come in this in natural format. So they actually engineer this antibody in such a way that they have a unique binding to the HER2 in such a way that it's so effective that a single antibody is much better than any of these in combination. So this is very important for doctors, right? Because doctors don't want to give the patient first a trastuzumab and then see how the response is and then give the next one. Or sometimes they give in as a cocktail together. But now we have a single antibody, biparatopic, which can bind to different domains in the trans. So I'm not going to go through these details. These are cryo-EM pictures at around 7 angstrom resolutions. But I just want to show the schematic how the antibody binds. This is a HER2 molecule in the green here. And you can see one part of the antibody binds to the one domain and the other one to other one's domain. So this is nice. And this don't usually occur in nature. In nature, all our antibodies, the Y-shaped structure, both of the V's bind to the same domains. And this is very nice. So what did we find? These are some of the data which I generated during my postdoc work. I found that like when I stain the cells with these antibodies and look under the microscope, I found that somehow this makes large caps or clusters. And this is very common. When you have proteins on the plasma membrane, and when you try to engage them with the antibody, they come together and partition to big protein islands because plasma membrane is very fluid. On the contrary, we also have something called small microclusters. These are like small patches. You can clearly see the difference in the microscope. These are like giant aggregates of receptors. These are like small patches of receptors. And if you see that the red one, which is actually the receptor caps, you see that mainly only for the Zani antibody, mainly at 5 and 15 minutes, and then it resolves. But you do not see that red picking up with the competitor ones, the TRAS or the PERT or the trans or PERT in combination. This was very exciting because this gave the first hint that it is behaving very differently. And we want to take a closer look because this was like just looking in the microscope and we don't know what is happening with the receptors. So this is where I was really involved in getting a detailed picture of what is happening to these clusters. And I want to please apologize to slightly switching the topic, and I promise you, like I go very slow. We have a problem in microscopy that when you use a light, you cannot distinguish two molecules that are very close to each other. We call it the diffraction limit of light. It's around 200 nanometers. So if two proteins are there, they're very close to each other, we cannot say they are apart or together. If it's far apart, it's good, but when they're coming together, we cannot do that. So when these small receptors cluster together in small nanometers, we need to use a special kind of microscope to break the diffraction barrier and visualize them. And one of that approach is called stochastic optical reconstruction microscopy, or called STORM. What you do is that if you put light on the object, it will light up everything, but the way we shine the object is not light up everything, rather it just lights up few molecules. And it lights up here, you localize that molecule, then you switch it off, and then you get another molecule switched on, then you localize the molecule, switch off. In that way, you can create a reconstruction of what is a pattern. To give you an example, this is something which you find in the very beginning of the reconstruction. You just get some dots because it is an image, you get some dots. Anybody can make any guess what this could be? I know, I put you on a tough spot. As the time goes on, we keep collecting the data and data and data, and you can finally get the reconstruction. So what we do is that, like, you know, if you try to shine light on the whole thing, it will be just a big blur. So we try to isolate individual molecules at a time, and we try to do this imaging for a long time, like 30 minutes or 45 minutes. And eventually you get a reconstruction of what that structure is. So this is what we have been doing, and to do this, we had to do in a very fancy microscope. I'm not going to describe about the details of the microscope, but all I want to tell you is that we have a way to do this in autopilot, where if you image for a very long time, because of the heat in the system and because of the drift in the table where you're imaging, we get very blurry images like this. But we have developed a very unique microscope in the campus with the help of collaborators, where you can see that they are completely drift-free. It's completely stabilized in X and Y in 2 nanometer space and Y in 6 nanometer space. You can really see how this image, the ground truth is something like this. And this is how I spend most of my days in the lab, in the basement, with all these lasers. There is no eyepiece or anything. This is like the most fun time I had during my research. And we still need a lot of algorithm to analyze the data. We cannot just collect all these dots and then say, okay, what is happening? So we need to have a very powerful algorithm. Again, I'm not going to go into details, but we have actually a very powerful method. Together with one of my colleagues who is a mathematician, we build this so that we can nicely isolate the clusters from monomers and so forth. So what happens to our HER2? So if I put the Zani antibody, you can see that, like, among all the things on the top, you see that these are actually giant clusters. And this is actually the heat map of the same data with the area color-coded. And you can see that these red indicates the very large clusters, and you do not see that in the, sorry, the point is not working. You don't see that in any of the data sets. So really to appreciate that, like, even that caps I showed before in that confocal, which was like that, we can go into the super resolution mode and find that these are actually giant clusters. So what does this all mean now for people for the cancer? These are data from the mouse. This is a xenograft model, the tumors. And I want to bring your attention just to the Zani antibody, all the one in blue. And you can see that, like, the tumor volume in the y-axis is all going down with the Zani antibody. You can see the competitor antibody TRAS or the TRAS plus PER is still not as good as other one. And when you keep the tumor uncontrolled with anything, you can see that it keeps on growing up. So we are trying to understand what these bispecific biparatopic antibodies do in terms of cancers, but we have a lot of proposed mechanisms, and this is a big scheme. And what we think is that, like, we're having these huge caps on the plasma membrane, and this causes a lot of differences in the way the cell respond. And you have the complement-mediated killing, which, again, I'm not going to the details. We have also studied these complements. We also have the killing mediated by the NK cells and also the macrophages, and we also have the changes in the signaling, because when you put an antibody on a receptor and they come together and they start signaling, the cell will actually put a break. But when you make these massive clusters on the plasma membrane with these bispecific or biparatopic antibodies, they put a big break such a way that signaling is all halted, thereby the cancer growth is reduced. So these are the ways by which we take a nature's protein or natural protein and how we engineer it to make it better. That was all about BiTEs and about antibodies, and slightly switching gears to cell-based therapies. And this is the last part of my talk, where I wanted to bring your attention to how we are engineering our immune system to make it better for us and kill the cancers. So again, back to some of the immunology basics. This is a T cell on the plasma membrane. You can see the T cells doesn't actually bind to any antigen by itself. It has to be presented through a peptide MHC complex. You can see it's a small peptide there. Whether it's a tumor or a viral or even a self-peptide. And you can see these are all the signaling domains. So the T cell by itself does not signal. You need to bind to the peptide, and then these are signals. And now we have the other component of immunism, the B cells. And the B cells have these antibodies or the B cell receptors. So what we do is that, like, we might now make a chimera. We take a part of the B cell and we fuse it with the T cell receptor to make a total chimera, which this molecule does not exist in nature. And you can see that this is coming from, the head part is coming from an antibody, and the signaling part is coming from a T cell, and you have some of the co-receptors there. By doing that, you have a single molecule which is called chimeric antigen receptor, or CAR T cells, and it can bind to a tumor antigen right away. So why do we do this? Because tumor has found its own ways not to present the tumor peptide. So when the tumor, every day we have mutations, and our immune system is alerted by every single nucleated cell. They actually present the peptides. Our immune system scans, identifies, okay, you're not mine, I'm going to kill you. But once you become more cancerous, the tumor has found very smart ways to prevent this presentation. But we can still circumvent this issue by getting a tumor antigen and have a totally hybrid receptor, something like a head of a horse and a belly of a lion kind of thing, and then you can have them still kill the tumors. This is how we beautifully take the systems of both the B cell and the T cell to make a total chimeric cell. So my current research is working with the CAR T cells. And I'm just going to skip on the generation because we are learning a lot and we keep on adding different domains to these receptors. But what is most exciting is like what happens to a patient who gets a CAR T cell therapy. So it is a nice schematic here. They come to the clinic, they get the blood drawn, and we get the T cells out. Once we have the T cells, we go to the lab, we engineer it. So we use a virus to transduce these cells, and we make them CAR T cells, just like the previous schematic I showed. Now they are fully equipped to kill the cancers, so we put them back into the patient, and they kill the tumor. I mean, this is a very simplistic explanation, but we have much more challenges than this scheme. But this is the whole idea. Like, the patient comes, if they have a very functional immune system, get the T cells out, engineer it, and put it back. I just want to bring the Emily Whitehead to your attention. She is one of the longest survivor of the CAR T cell therapy. This paper came actually last year, 2022, in January. A decade-long leukemia remission because of the T cells, CAR T cells. And you can see that after 10 years of infusion, you take the blood of the person, you can still see the cells which are CD4 positive. All these dots you see here are what you gave to that patient 10 years ago. And you can still find that they're still active and still doing it. But something new what we learned is that the cells which are killing the tumor, because you cannot just kill the tumor and then go silent. Again, the tumors come and you keep on killing. You need persistence. The long persisting cells actually is something called like CD4 T cell or CD4 CAR T cell. And this is changing the way we think about immunology, because some of you might remember again, we have T cells coming in two flavors, a CD8 T cell, which is called a cytotoxic T cell, and a CD4 T cell called a helper T cell. So helper T cell just secretes cytokines, and it's like a master regulator of an orchestra. Tell them, like, okay, you go and kill that. You secrete cytokines. It's kind of regulating everything. But CD8 is cytotoxic T cell. It scans, finds a tumor or a virus, kill it, go to the next one, kill it, and go on. Surprisingly, we are like, this is not textbook information. If you go to any classical immunology textbook, we will learn CD4 as a helper cell, CD8 as a toxic cell. Now we are seeing in the patient that after 10 years, somehow the CD4 is persisting, and they are still having cytotoxic characteristics. So this is a time like, we are trying to, okay, what is happening? We have to change all the information we have in textbooks. So this is very exciting data sets coming from that. We have many roadblocks in the CAR T cell therapy. Just for the sake of time, I'm just going to go it very quick. It's much more expensive. We have a cytokine release syndrome for some patients because of that massive cytokine production. And it's very, very challenging with one donor to patient ratio. And this is where exactly the company which I am working in comes into picture. So just completely transitioning to my current work in Vancouver in Notch Therapeutics. So our aim is now to make a cell-based therapies applicable to everybody. It's not that the patient comes, we take the patient's T cell, go to the lab, engineer it, expand it, come back and put it. I can take my T cell, I can engineer it, and I can give to anybody in this room. So this is the way we are thinking. How do we do that? To do that, we have to first make an induced pluripotent stem cell. Just again, this schematic is from a patient, but what we do is from a healthy donor. So we know that all of our cells in the body are fully differentiated. Our skin cells, our liver cells or other cells, they're all differentiated. But we have 4 factors, which is called differentiation factors, called Yamanaka factors. If you put into the cells, the somatic cells, they reprogram. It's like going back in the, a time machine and become a stem cell. We call that stem cells induced pluripotent stem cells. And this is very exciting because after this, just four factors can completely make your fully differentiated skin cell become an induced pluripotent stem cell, and you can make a liver out of it, heart out of it, kidney out of it, whatever you want. So fully differentiated cell going back and redifferentiate another cell. The most exciting thing is that Yamanaka got the Nobel Prize for these 4 factors in 2012, the Nobel Prize in Physiology and Medicine. But the way we are looking at this induced pluripotent stem cell is we can take these cells, we can engineer them to make CARs, and then we can make them become a T cell or a natural killer cell or a macrophage, whatever we want. So this is exactly what I'm working on. So we actually take healthy donors, get them into an iPSCs or induced pluripotent stem cell, engineer our cells thoroughly, so much, in such a way that we make it a all off-the-shelf car product and give it to anybody. And the most challenging part of this is, I told about the reason why we're doing this. Like, the conventional one is one patient at a time. What we are doing is from one donor to many people at a time. So it really brings down the cost of the product. This is about my company and other details. I'm going to skim through that very quick. But the most exciting thing is that, like, one question you might be having is about immune rejection. So we actually edit our induced pluripotent stem cells in such a way that it evades the immune rejection. So this is very important because, if I take my T cells, any of my cell, whether a T cell or a B cell, I make it into an induced pluripotent stem cell, and I make it for a, say, B cell lymphoma or some other tumors, and give it to anybody here. The first thing your body does is to reject it right away because it's foreign for your body. But we edit our cells in such a way that it hides in the immune system. Both the NK cell and the T cell response, natural killer cells and T cells, is completely hidden. We also put a kill switch into our product so that if we think our product has gone crazy and become cancerous, the clinician can give a pill and the product just dies off, or the cell just dies off. So that is also engineered. So the way we are editing our product genetically is such a way that, like, we are playing God. So we are editing it to an extent it hides in a patient's immune system, kill the cancers. If that goes crazy, you put a pill, kill the product. So we are racing towards this goal, and there is a lot of obstacles to come. Especially the immune rejection is a very big challenge. So our aim is to make off-the-shelf product so that, it's a medicine. You can go to, when you have cancer, you can go to a bank, pull up some cells from liquid nitrogen, and then you can put it into your body. And that is the kind of way we're looking forward. These are some of the important parameters about proliferation, stability, and potency. But I want to show you one video, and I'm so excited to show this because some of the data sets which I'm showing now is actually from my work, which I done like probably 3 months ago. So I want to show you what is this figure is. This figure shows that you are actually challenging the product or the T cells, the induced pluripotent CAR T cells, over multiple rounds of killing. And that's very important because you need to kill the cancer cells again and again and again. And what you see here is a tumor cell area. And all of this line shows that there is this is just a target cells, means only the tumors. The tumors keep growing. Tumor cell area keeps growing. You can see the tumors just keep growing. But when you put the iPSC CD8, that is our product, or a primary cell, which is primary cell sense, in the sense that it is a bona fide T cell, which is it's a CAR, you can see that they both kill nicely. You can see over multiple rounds. And this is a fold expansion of the product. You can see that, like, these are two of our competitors currently. One is in the US. Both are US-based companies. But you can see that their fold expansions at this range, we are getting like a massive fold expansion of the cells after all these rounds of killing. And if I were to play that movie, everything in the green is actually a cancer cell. You can see that over the time, the green keeps increasing because they keep dividing and dividing and they keep going up. And that is just uncontrolled tumor growth. And what I want to bring your attention is to this movie here, where is our product there. And when you add our CAR T cells to there, you can see that in a short time, they identify the cancers, they become a little clump, and they kill it off, and they proliferate. So this is a real demonstration of, at least in vitro demonstration of, like, you can see that the complete clearance of this happened, and there is no green left over. And this happens in multiple challenges too. So the way we are trying to build our company is that, like, we want to make this into we are currently in the preclinical phase. As I speak, the testing is done in the mouse now. And I heard that's very exciting data coming up. So the mouse were actually having the tumor. And now after putting our product in the mouse, it's nicely reduced. Tumor volume is nicely reducing. And we are giving much more challenges to that. But our aim is actually to go from a preclinical to clinical phase and also commercial phase. We can do this in large bioreactors. So this is exactly what our technology has now accomplished us to make very less expensive, consistent, homogeneous product so that we can use for anybody. We have a lot of things to accomplish, but at the moment we are currently in as I told you, in the functional T cell part where I show these videos. And in the early phase. So we need to have a large manufacturing, and eventually one day hopefully, if everything goes well, in this hospital, you'll be using some of these products. I told all the good things about CAR T cells, but I should also caution you about what are the problems or main challenges we face. And this is just my last slide to bring down the discussion. We always have CAR failures happening. And the main reason is that we cannot harvest enough cells from the patient. And this is why exactly, as I told you, sometimes our product is superior to the patient samples because patient might be in his 70s or 75s, and then his immune system is so weak, we cannot get enough cells, engineer it. So this is a failure. So this CAR failure in the clinic is something which we can actually circumvent because our product is coming from an induced pluripotent stem cell. The antigen modulation, where you actually know cancers actually keep on changing. The cancers are having very heterogeneous expression of the tumors, and this keeps changing. This is a problem we have. And we have to understand that you give one product and it maybe kill 90% of the cancers, but you have a residual 10% which actually keep on growing and so forth. So this is something is we had to work around. Toxicity is actually one of our major problems because we want to have a product which is not only killing the cancers multiple rounds and proliferating, but very specific and safe for a large population. So this is all coming in that editing part of the cell where we are really making sure that it hides in our immune system, in anybody's immune system, and thereby continuously killing all the things. And obviously we have challenges to address solid tumors. Even though the T cells can infiltrate into solid tumors, the tumor is a cold environment. The tumor secrete factors which prevent the T cells from getting a response. They go to the tumor, they find there and this pause. So this is something which we had to address. We had to find ways by which we can make these tumors much more better to address the solid tumors. But some of the challenges are very real, and even if it works excellent in the mouse or even in the phase 1 trial or phase 2, we still have a long way to go on. And we had to really monitor what happens to the product after 10 years. Just like the Emily Whitefield story I showed you of 10 years of persistence. We hope that maybe in another decade or so, we can show our product also is there in the patient for a very long time. With this, I just want to conclude my talk. I have a lot of people to thank. Basically, the 2 companies I worked for, Notch Therapeutics for the CAR T-cell work and Zymeworks, and all of my collaborators and professors, especially the people from UBC Maths and Chemistry, for building these fancy microscopes and helping me with the analysis. And this, I want to thank all of you for your attention, and I'm happy to take any questions if you have.
Thank you, Libin, for that wonderful talk, even though I didn't understand much of it. I'm sure there are people who understood your talk. And I would request Chepsi to make a comment and ask some questions if you have.
Thank you, Dr. Libin. That was really fascinating to hear your approach to CAR T-cell therapy, because I work in hematology, and we all recognize one of the challenges, at least in India. One is the simply prohibitive cost which comes with CAR T cells, and it is really sad that not a single major company has initiated a single trial in a country which has the largest amount of patients. That is one, and we believe the regulatory roadblocks is one of the reasons that happens. But your last approach that you showed, that you're trying to reverse engineer, you're doing this iPSCs and use bioreactors, probably that is a scalable model which should reduce the cost. But my question to you is, one, with all these CAR T cells, it's very clear that we are targeting B cell dominant malignancies or solid tumors. And you have also created a system where you want to evade the immune system by doing that. So in the future, how do you think it's going to pan out? And I also noted that you selected out CD8, whereas you realized that the longevity is through CD4. So what is your thought process into why you chose CD8, one? And second, going forwards, how do you target other malignancies? Because by default, we do have good therapeutics available already. It is the other fields which need this magic more. I'm not belittling the importance. And third, there's Emily Whitehead story. We all know the success part of it, but there is also the patient who didn't make it. So what are the lessons that you have incorporated from that?
Yeah, I mean, all fascinating questions. I'll try to answer all these one by one. The first question you asked was, what was the reason we went for the CD8, even though we know from the clinical data from Emily Whitehead that CD4s? It's mainly the FDA. So when we are making this product and when we are going to FDA will have a very rigorous look. What is your product made of? Because any induced pluripotent stem cell after gene editing can go really crazy. So we want to make sure that stemness is completely gone and it is completely differentiated into a T cell, and tomorrow it won't become, say, a macrophage or say a dendritic cell or something else. So we want to show that as the process of differentiation goes on, we complete it, and we want to market the product as a CAR expressing CD8. That said, much more recently, we have been seeing data from other companies or competitors where they are not really calling it as a CD8 product. They're calling it as a CAR product, iPSC-derived CAR product. It has features like an NK cell, like a natural killer cell. It has features of a CD4 cell. It has features of a CD8 cell. So the cell is not fully lineage committed into a CD8 or a CD4 or an NK because it came from a stem cell, and we are less. Whatever happens in the thymus, bone marrow and thymus, we are doing in a Petri plate just by giving factors. So it's very complicated, and you're right. So one of the main reasons we use CD8 is because just to make sure that a product which we have as CD8 can really do that. And something which I did not show is after 4 rounds of stimulation and killing, they lose the CD8 because it's just a co-receptor, and the cell is more like receptor-less, like it lost the CD8. So it's like a double negative cell, no CD4 or no CD8. It's a CAR-expressing lymphopoietic cell coming from an iPSC. It's still able to kill multiple rounds. But that product, we cannot take it to FDA. FDA will ask you, okay, what is your product? That's the challenge, and we are still trying to address it. We wanted to make lineage committed into either a CD4 or CD8 or NK. That's the first part. Second part, regarding the cost, you nicely addressed it. Yes, it's very expensive, and that is mainly because when the patient comes, you need to get their blood, get the T cells, and all this done in a GMP facility, and you need to engineer these cells, expand these cells, and then give it back to the patient, sometimes even multiple infusions. This is a really cost-expensive process, and there are also potential risks in that too. And that is why, as you rightly pointed out, we are using the induced pluripotent stem cell so that any donor can actually, you can take the cells from any donor, make an iPSC, and then you can edit it and then make it into that. So our aim is to bring down the cost. Because if you have one good healthy donor, we have one good product, it's a homogeneous product, it kills that cancer, we go for it. Addressing the third part, and you nicely pointed out, yes, we have good products for the B cells, so CD19, or the B cell lymphomas. Much more challenging is the other one. And the reason we are doing this is because we have to have a proof of concept to show that it works good in suspension, like tumors, not solid tumors. If we have a good product which can demonstrate good cytotoxicity and proliferation, and multiple rounds of killing and persistence without cytotoxicity in the EC system, to be very honest, then we can say that, hey, look, it really works well. Now we can go more challenging ones. And this is actually the good thing, because one thing I didn't address is the solid tumors, where the best way currently people are doing the solid tumors is based on something called TILs, or tumor infiltrating lymphocytes. When you do the biopsy of a solid tumor, you get some T cells there. And these T cells actually homed into that tumor with the aim to kill, but they are, again, as I told you, like paralyzed because the tumor environment is very cold. Tumors secrete factors which prevent the T cells from getting fully activated. So what we do is at the part of biopsy, you actually understand what the cancer is, but you take those tiny amount of T cells which infiltrate in the tumor, go to the lab, give IL-2, give a lot of growth factors and expand them, and give a small dose of chemotherapy and put it back into the patient. And TILs, tumor infiltrating lymphocytes, have been shown to be so far quite good for this solid tumor approaches. CAR is still challenging because how much we educate, it's going to be challenging. So why did the tumor infiltrating lymphocyte go into the tumor? We still don't know. It just happened by nature. But we can exploit them, expand in the lab, and put it back into the patient, and without any major complications. That's one thing. So yeah, I think all the points you raised are very valid about why are we targeting the suspension cancers now, because we want proof of concept. We can bring down the cost by using iPSCs. The bigger challenge for us is really like defining, are we really making a product which does not in another 5 years become something else? because that is very important. When all this maturation happens in the thymus, that they are lineage committed CD8s or CD4s. But when you do all of this in a lab, giving all of these factors, literally it's like everything happens in bone marrow, and in a thymic setting, you're just replicating everything in a Petri plate. So yeah, we have learned a lot how to do that. We have a lot of patents and how to do the tricks, but that's where we are heading at. I don't know if I answered all of your questions. I think the cost I addressed, the reason of using the suspension cells I addressed. Did I miss anything?
No, you answered quite elaborately. Just off the thing, when you use these Yamanaka factors, does any part of the royalty goes to him?
Oh, I don't know whether the factors are patented, but we also use additional factors for that. So yeah, obviously Yamanaka, I think, patented that, I think. But all patent expires after 10 or 15 years, and then we can go. This was, I think he patented back in 2007 or something. There was a Cell paper in 2007 when he had, if I'm not mistaken, he patented this, and then I think it's there. I am not very familiar with the gene editing part, because they are having the most hardest time in our company to do all these edits to make the cell completely like a shadow in the cell, and just identify it. Because yeah, we can do that, because the immune system, our immune system recognizes our own cells and foreign things based on different receptors, as displayed by our cells and also other things. So we can actually edit them, and we have some good success with T cells, but NK cell is the toughest part.
Thank you, Dr. Libin. As much as it is exciting, I think it is also very scary.
I know.
Because to me, after this talk, I don't think the scenario in No Time to Die, the Bond movie, it is not too far away.
I know.
See, I'm an endocrinologist. What I'm more concerned and worried about is that once this immunotherapy and the checkpoint inhibitors came, we are facing a problem of immunity, in the sense that we cannot take our immunity very lightly. Because we see a lot of central hypothyroidism, central pituitary problems, because these medications that we use, mainly the MABs, the monoclonal antibodies and all that, or even the checkpoint inhibitors, they create an autoimmunity against the thyroid, against pituitary. So with these kind of therapies, we cannot ignore the fact that immunity can be turned around in a different way. And when you say Emily's story that it can stay in the system for over 10 years, that's really scary for me. And also,
it happens with our normal, for example, since we got the COVID infection now, our memory T cells will be there for a very, very long time. And same for the B cells as well. So,
true, that's why we are seeing a lot more of heart failures with no other cause other than vaccination. We see a lot of new different diseases in people whom we do not suspect it at all. So there is much more for us to know about immunity, and not just look at the exciting part of it. We have to always be careful of that. And also we are waiting for the day when you can actually make a pluripotent stem cell into a beta cell. For over 10 years we are still waiting for insulin production.
That's a good point. I'll address both of it. The first point regarding the autoimmunity, actually you brought up a very valid point. We know that our body has something called central tolerance. So that the way our T cells or B cells came, or T cells came out of the thymus or the B cells of the bone marrow, is that they have an education to spare our own cells. And somehow this tolerance is lost over age, first thing. And second thing is after some infections. We don't fully understand why. Even after COVID infections, we are seeing a spike in autoimmunity in people who were never autoimmune before. And even this is reported even in people with mild COVID or even moderate COVID, not even severe ones. Obviously, severe ones had the worst outcome. But these are things which you do not understand. I totally agree. So, but I'm not aware about biologics like antibodies itself cross-reacting with organs like the pituitary or anything, because that is where probably the bites become more better, because the whole antibody, there's a Y-shaped structure and you have this identical binding for both of the Fab domains and an FC part. Bite is like fully engineered with taking only the caps of it and putting it together. So in that way, it's a little more smarter to be more specific and to prevent off targets. And the reason I told that is because in biology, especially antibodies, you have this FC part of this thing, right, which can bind to any FC receptors, which is on APCs or other cells. So this is a challenge, and I acknowledge the point you're saying, but I think some of these can be circumvented by actually bites, because bites are like extremely small molecules generated in such a way for the whole purpose of reducing these risks. And the second point, ma'am, which you told before, keeping the mic, sorry. Beta cell, yeah, sure. That's a very valid point. Yes, we wish we could actually engineer them. The thing is that, like, when you make an iPSC and then you differentiate them into all different kinds of cells, they all require different factors, and we do not have fully uncovered this completely. We have a pioneer in our own company. Her name is Deepika Rajesh, and she has been a queen of iPSC differentiation. She has made all kinds of cardiomyocytes and all the different kinds of cells, more than 20 patents for herself. But I would have probably to ask her whether she has any colleagues or other people who might be working on the beta cells, because this is a true thing. Like, people who actually would benefit the most might be, that. I think the biggest challenge is we do not fully understand that ontogenic process. Like, what are the signals you need to give at what time, at what threshold, because everything happens in our naturally as a baby or something. We are recapitulating in a Petri plate. So there are genuine challenges. I agree.
More precise, because of the artificial intelligence guided nanotechnology, delivering the molecule to the specific site will be.
That's true. That's true. Because the company which I mentioned, the AbCellera, which actually came with the first COVID antibody, they actually have a platform not only to discover the antibodies, but they actually use an AI-based approach to fine-tune the antibodies. Both, they can actually run it through their multiple iterations and see what is the structure of these bispecifics and how exactly they work. So yeah, there are a lot of companies who actually use AI to predict better outcome, and they have been showing that.
If there are no more questions, we'll conclude the talk. Thank you so much, Dr. Libin, for that wonderful session. I would have titled it Playing God, Pros and Cons of It.
Yeah, I agree.
That was an amazing talk. And so we really thank John Valliattu for bringing you over. We look forward to having you for many more sessions. So can we all give a good round of applause for Dr. Libin for coming all the way from Canada and presenting to us the art of playing God.
That's true.
So have a wonderful day, dear family. Thank you all.
Thank you.